This presentation is about recent advances in Corpus Callosum dysgenesis genetic research. Okay. Here is what's coming up in this presentation. I'm going to present our group, the goals of our research, and the progress that we've made until now, and where we're headed in terms of genetics, genotype-phenotype correlations, and prediction analysis. We are located in San Francisco, and we run the Brain Development Research Program at the UCSF School of Medicine. Here is an overview of our team. On the left side, people that are at UCSF. Dr. Elliot Shear is the founder and is leading the Brain Development Research Program, or BDRP. He's a professor of neurology, pediatric, and genetics. And my name is Emanuela Argilli, and I've had the pleasure of working with Dr. Shear for the past almost 9 years, and then especially on dysgenesis of the corpus callosum in the past 5 years. So other members of our team that I'd like to acknowledge are Dr. Elizabeth George. She's a neuroradiologist, and Caroline Lee, our clinical research coordinator, Gilberto D'Agente, our bioinformatic analysis, Ruben Thomas and Min Shin for statistical analysis. And then outside of UCSF, we work closely with people like Dr. William Dobbins at UMN, and Dr. Lynn Paul, who you just heard talking from Caltech, and Linda Richard, also you just heard from Washington University, as well as people at the Broad Institute of Harvard and MIT, and the exome sequencing company GeneDx. And our founding comes from many different sources, but especially NIH, UCSF itself, the March of Dimes, and many individual donors. So thank you. The goals of our research are to identify the genetic causes of disorder of brain development, in particular of CCD, Corpus Callosum dysgenesis. We use imaging and genetic findings to identify at-risk children and those that are likely to benefit from early intervention. We strive to provide families with information for genetic counseling, and we sometimes run biochemical and biochemical models as a first step towards targeted therapies. Here's just starting from the basic. The corpus callosum is the white matter tract connecting the two hemispheres of the brain. It is important in integration of motor and sensory information, as well as in higher cognition that is associated with executive function, social interaction, and language. It is located deeper in the brain, and it's as shown here, the fibers and in the DTI map, the fibers are primarily left to right oriented, and then they fan out more laterally and intermingle with other projection. So the corpus callosum is a brain commissure. So there's not the only one that humans have. We have 9 commissures. So those specialized white matter bundle of fibers or that connects regions of the two opposite hemisphere of the brain. So, but here in this map is to show you how big the corpus callosum is compared to the other commissure that I'm listing just the first 5 here. The corpus callosum contains 200 to 300 million axons and is in green here. And the second biggest one is the anterior commissure that only has 7 million axons so much smaller compared to corpus callosum. And here, the anterior commissure is located below on the anterior part of the brain, and it's very we pay close attention to the anterior commissure, but I'm not sure if I'm gonna get talking about that later. So corpus callosum dysgenesis is one of the most common congenital brain malformation with up to 1 to 2,000 live births frequency. So the three main types that we include in our study are complete agenesis. So as shown here, so there's complete absence of the corpus callosum, partial agenesis, which typically the anterior portion of the corpus callosum is preserved, but is a shorter corpus callosum, especially in the posterior aspect, or more typically in the posterior aspect. And then hypoplastic corpus callosum is a corpus callosum that is thinner than normal. The etiology of CCD is mainly genetic. So it's a quite complex picture, as Linda hinted about earlier in the first talk. And up to date, there's 900 genetic syndrome that include CCD in their description. And CCD genes span every chromosome. I hope, should I stop and see if everything is okay? Let me stop for a second. Is everything okay? I see a lot of movement. Okay, that's good. I want to keep going then. Thank you. Okay, so go back to presenting. Okay, so sorry. As I was saying, and there's so one of the main causes of CCD is genetic, but there's also ongoing work to unravel further and but less common causes, such as uterine infection, injury, and toxicity that happens around the 11th to 22nd weeks of pregnancies, which is when the corpus callosum forms. So genetics of CCD is what we're going to talk about. And so we're going to talk about the DNA, which is contained in, so the genetic code or genome is contained in every cell of our body, and is made out of DNA. And the DNA is made of 4 basic chemical units that are abbreviated with the letter A and T and C and G. And a gene is just a small piece of DNA that contains information to make a product. And this product is a protein that has a specific function and many specific function in our body. So we go from the genome, which is 3 billion sets of these genetic letters or nucleotide. And one of the function of the protein is to form the human brain, which is super complex and with 100 billion neurons and 100 trillion synapses, which are the connection between neurons, so how the neurons communicate with each other. So as you can hint where I'm going to, how complex the molecular mechanism for CCD are. So CCD is a very heterogeneous condition that results condition like anatomical change that can result from the disruption of one of many important steps in the normal cerebral development. And so, so 5 of the main steps for a normal brain development are neurogenesis, or corpus callosum development are neurogenesis, so the making of neurons themselves, midline formation, so the division of the 2 separate hemispheres, neuronal migration, so that each neuron goes to the right place, as well as axon guidance, which are the longer, the extension of those neurons that make the corpus callosum need to go across the midline and go to the opposite side, as well as post guidance development. So many different places where the normal development can be disrupted and cause CCD. So so we use this technique called whole exome sequencing for sequencing only the protein coding regions of the DNA. So that is only 1 to 2% of the entire human genome. So other regions are called introns, and those are we don't sequence in whole exome sequencing. So the genomic variants, or what most people call mutations, are the difference in a person's DNA compared to other people's DNA. And researchers have made up a reference human genome sequence based off of many different people's DNA to help to compare to every single new exome that is sequenced. So every person's genome is about 99.6 identical, and therefore 0.4% different to the reference genome. And our job is to identify only the small subset of genetic variants that might lead to disease. So, so the output of this whole exome sequencing is just the raw DNA nucleotide sequence that we compare to this reference genome that we've talked about to identify this error or variance or mutation. Some variants are very small changes, so one single nucleotide change. So if this up here is the, hopefully you can see me, is the reference genome. It should, in this position, there should be a C, instead it's a T, and so that is called a single nucleotide variant, or SNV, or the changes can be a little small insertion or small deletion that are smaller than 1,000 base pair. Those are called indels. So in this example here, those two, the A and C paired with the T and C are missing, and in this other here to the right, they are inserted. Another type of variant is copy number variants are instead are larger deletion or duplication in the genome that might usually typically affect more than one gene. So what's the impact to attribute a value to this variance or mutation on human health? We look at them. First, we look at the functional effect. So what does this change in the DNA do to the protein that is supposed to produce? So, and that effect can vary from it doesn't do anything to the protein, so normal function of the protein, to, on the other extreme, abnormal function or a complete loss of the function of the protein. But how does, and the second thing that we look at is how that change, how that effects translate into health. So what's the clinical effect of that variation of that mutation? And that again goes from we're not sure of what effect it does to it's clearly pathogenic or disease causing. So for the clinical significance of every variant, we follow the American College of Medical Genetics guidelines that offers a 5-tier terminology system. So the impact of each variant is classified from being benign, so no effect on human health, to likely benign, likely no effect, to VUS, so variant of uncertain significance, so we're unsure about what the effect is on human health, like to likely pathogenic or to pathogenic. So definitely, or with pretty good evidence, those changes are disease causing. So we strive to perform trio exome sequencing, which is not only sequence the DNA of the person with CCD, but we sequence also their biological parents when available. And to do that, we collect typically saliva, or it could be any other tissue like blood, body fluids, or biopsy. And then in UCSF, we'd extract the DNA. And then we send it for sequencing to our partner at the Broad Institute. And then with the output, the raw data comes back at UCSF when we interpret the results. So we give to each variant, we classify as, benign or VUS or pathogenic. So what does the trio whole exome sequencing allows us to do is to enable identification of de novo variants, so the variants that are not present in the previous generation. And those are spontaneously arising variants have a very significant role in neurodevelopmental disorders. So every person has about 40 to 80 de novo single nucleotide variants, so the small changes that we talked about before. So every one of us. But in the protein coding region, so in the exon part of the DNA, we only have 1 to 2. And most of those de novo variants have no effect on human health, so are neutral in impact. So, so I'm gonna highlight some of our results that comes from the enrollment of people with a CCD for the past 20 years by Dr. Elliot Shear, and for which we have completed all exome sequencing, and there's been a complete interpretation of the data. And so we have, our numbers are that have individuals that have completed whole exome sequencing and for which each variants have been interpreted, and those are 700 individuals, 500 trios, and about 200 duos or probands only. So we try to promote gene discovery, so find new genes associated with CCD, and we perform the analysis 2 ways. One is a cohort-wide analysis. We only look at all the de novo variants, and we try to see if there is a difference between the de novo variants that we find in the CCD cohorts versus the de novo variants that are found in a neurotypical control group. And so this analysis, we find that 54 CCD risk gene, including 10 that were not associated with previously with CCD. And then secondly, we perform the analysis case by case. So on an individual level, we look at every type of variants, not only de novo, and includes all the also larger duplication and deletion. And this allowed us to find even more candidate genes for CCD, as well as to identify features that correlate and to correlate and try to predict a certain clinical outcome. So the first part, the cohort-wide analysis of only the de novo variants was performed in a cohort of over 1,000 individuals with CCD and their biological parents, which is one of the largest trio whole exome sequencing study of CCD to date. So, and then about 500 individuals were from a collaboration of UCSF, UMN, WashU, and Caltech for research whole exome sequencing. And in addition, we obtained 608 data from 608 individuals with CCD from GeneDx, which is a clinical whole exome sequencing company. So as I mentioned before, so this we were able to so if you look at this map, this table here, on the x-axis, you have all the different chromosomes, so 1 to 22, and then chromosome X and Y, and then every dot represent a gene. And if a gene is above this red dotted line, it means that we have found that people with CCD have are enriched in variants in these genes, and therefore we consider this gene candidate genes or risk gene for CCD. And of these genes, 44 were already known to be associated with CCD, but 10 genes were not previously associated with CCD, so we consider those candidate genes. The second part of the analysis is a case-by-case analysis of every individual, of every family. This is only in the cohort, in the research cohort from UCSF, UMN, WashU, and Caltech, because we not only needed a comprehensive variant interpretation, but full clinical history available, as well as MRI available for internal review. And also we needed to confirm the CCD diagnosis by our neuroradiologist team, and we only include in our cohort complete agenesis, partial or hypoplastic corpus callosum, while we're not including like a hyperplastic corpus callosum or just dysmorphic corpus callosum. So that those criteria, with those criteria, we have a cohort of about 471 individual, where we have all this information available and which have completed and whole exome sequencing, and that has been fully analyzed. So this is a breakdown of that cohort. We have 48% has complete ACC, 18% partial, and 30% hypoplastic. 44% is female and 56 male. And that cohort, there's 20% of people are kids with less than 3 years old, and 80% are older than 3 years old. So the overall result that we were able to find a positive genetic diagnosis in 56% of the population, which is here is 47% are positive in known CCD genes, and then here 8.7% we have a positive genetic diagnosis in a gene that was not previously associated with CCD. So those are like novel genes. I think I wanted to point out that 6.6% have a VUS, which are very high priority variants for us, and we're like really trying to attribute the correct value of the correct interpretation to these variants or mutation. Are they pathogenic or are they benign, basically? So the advantage of performing trio exome sequencing is that we have a higher yield of genetic diagnosis. So we were able, as I said, to solve 56% of the cases where when a person with CCD and their biological parents are present and available for sequencing, versus 42% when only the person with CCD is available for sequencing. So I'm going to talk about genotype-phenotype correlation. Before I do that, I want to talk about how we classify corpus callosum dysgenesis in a little bit more detail. So up here in red is the complete agenesis. So absence of the corpus callosum. Here are two examples. I think that not much to say about these other than we take a look at the anterior commissure that is this like white dot here, and it's pretty normal or large here, and it's very small here. And then in the middle row is what we consider partial agenesis, and it can be sometimes there is only a remnant or like severe partial agenesis when only like a button or a little white dot is present. Typically, it's towards the front of the brain, so more anteriorly. And then classic partial is where the corpus callosum is short. So typically, the anterior portion of the corpus callosum is preserved, but it's very short. But the back, it's not fully, not all the segments are there. So, and then what we consider hypoplastic is a different presentation. So the corpus callosum, I mean, is thin throughout with no gradient, or can be thin and short with no gradient, or it can be just short in the posterior aspect, so towards the back of the brain, while the anterior portion is normal. Or occasionally, this is a little bit more rare, it's reverse hypogenesis, where it's thicker in the back and thinner in the anterior aspect. So also, I'd like to, different groups have this different terminology for this, but we consider the corpus callosum dysgenesis isolated when it's the only brain malformation present, and corpus callosum plus when it's associated with other cerebral anomalies. So if those cerebral anomalies are the ones in red, then it becomes a CCD plus for us. So those anomalies are cortical anomalies, such as gray matter heterotopia, or abnormal sulcation, like this example here has a cortical anomaly, or cerebellar brainstem myelination anomalies, or microcephaly. So in this example, there is not only a thin corpus callosum, but there's also a small cerebellar vermis and a small brainstem. The pons of the brainstem is small. There's other malformations that are sometimes seen with CCD, such as macrocephaly, white matter volume loss, interhemispheric cyst, lipoma, or ventriculomegaly, so enlargement of the ventricle. But those in our classification don't make it become CCD plus, because the clinical outcome associated with those malformations is less clear. So here, so now we have six different categories or types, basically: corpus callosum, complete isolated, partial ACC isolated, hypoplastic corpus callosum isolated, and then the same three category, but without additional brain malformation. And those are the numbers that we have for each cohort. So as I said, as a whole, CCD, the CCD cohort has a positive genetic diagnosis in 56% of the cases, but that varies depending on the brain anatomy that we observe. So corpus callosum, complete or partial agenesis have the lowest genetic yield, 42 and 41%, and the highest is hypoplastic corpus callosum, 79 when isolated, and 77% that we were able to solve with a genetic diagnosis in case of hypoplastic corpus callosum with other brain malformations. If we solve 56% of our cohort, that would be 264 pathogenic variants that we have found. And here is to show the heterogeneity of CCD. We found that only 81 were recurrent genes, so the genes were mutated in more than two or more individuals, while there were 164 unique genes or CMV, so that it present in only one individual. So there's many different genes that are associated, can be associated with normal corpus callosum development. So this is a list of our top CCD genes where we have recurrent genes in more than two individuals. In terms of inheritance, 81% of those were de novo, so not found in the previous generation. So absent in the mother, absent in the father. The mutation arises spontaneously de novo in the child for the first time. 11% are inherited from one parent, and 88% are inherited from both parents. I'm going to talk about inheritance pattern a little bit. There's three main patterns of inheritance: autosomal dominant, autosomal recessive, and X-linked. Autosomal dominant means that it's not on a sex chromosome, so not in the X or Y chromosome, but in one of the other 22 chromosomes. And dominant is when so we have two copies of each gene. So only one copy in this case is mutated, and that is enough to be causing a phenotype. In recessive, both copies of these genes are required to be affecting a phenotype, and in this case, CCD. And X-linked, they're on the X chromosome, so when previously I said that 11% were inherited, sometimes those inherited are from inherited in a male that only has one X chromosome. They are inherited from the mother. So the mother has no phenotype because she has two X chromosome, and the boy only has one. So that one with the plus the variant can be creating a phenotype, or CCD in this case. In terms of what type of variants we have found, so 20% are C and V, so those copy number variants that are larger pieces of DNA that are duplicated or deleted or missing, and those are typically affect more than one gene. So the rest, 80%, are monogenic mutations, so only affect one gene. And as in 41.3%, those variants are called missense, meaning it's a small change that cause the protein to be a little bit different. So we call them protein changing variants that have might affect the protein function. And then 31% have a protein truncating variants, but that we think can lead to a complete loss of function of the protein. So gene discovery from more than 700 CCD individuals. So while we're working on putting all these results together in one publication, we contributed to many different novel genes for CCD that have been, that have been discovered in the past few years. So we've published 9 of these novel syndrome associated with CCD in the past few years. And then we have many more that are in progress right now. And we also contributed many genes to many, some participant to the expansion of the phenotype of some previously known syndrome. So I'll try to move quickly because we lost some times in the beginning, but the next question is, do MRIs and genetics help inform clinical outcome? So the answer is yes. In his example is the different types of corpus callosum anomalies that we see, and how that relates to or is associated with the degree of global developmental delay. So so let's take the first example here. This is complete agenesis of the corpus callosum. So if you have in our cohort, complete agenesis of the corpus callosum is associated in 73% of the cases with no to mild global delays, and in 27% as associated with more moderate or severe global delays. So similarly, for partial ACC isolated, 72% of kids younger than 5 years old show no to mild global delays, and 28% show more moderate to severe delays. And that situation is completely reversed if you look at the other end of this this list with hypoplastic or thin corpus callosum with other brain malformation. We see more moderate to severe global delays in 96% of the individuals versus only 4% with no to mild delays. Here is this graph shows how a genetic diagnosis also changes is associated with different phenotypes. So again, if you take the first 2 bars here in the instance of complete ACC, so you have complete ACC with no genetic diagnosis found, so we were unable to find a pathogenic variance. So those cases are associated with only 7% of more moderate and severe global delays. Versus if we have the same corpus callosum absence, so complete ACC with a genetic diagnosis, those individuals tend to have more moderate, 52% tend to have more moderate to severe GDD. So a huge difference here, and that is a little less significant in the case with hypoplastic corpus callosum plus, because there's just less of those milder phenotype in that type of brain anatomy. So here's to look at other neurological feature observed with CCD based off on the brain anatomy. So looking at epilepsy or spasticity, for instance, and how that is different from one type of CCD and when it's associated with other brain malformations. We are also asking the question, what brain feature is more likely to correlate with a certain outcome? So in this case, epilepsy. And given that absence of the corpus callosum is a reference, so if complete agenesis is a reference, an additional cortical anomaly on top of that increases duration of epilepsy of 2.93 times. And cerebellar anomaly also significantly increase the odds ratio of developing epilepsy, while other brain features do not, the ones in gray do not affect, they do not add an increased risk of developing epilepsy. So same things we do with different outcomes. So here's to look at spasticity or CP. So here's the other brain features that increase the risk of developing that phenotype, and the ones that do not have an effect on that phenotype. Here's what here's other outcome associated with the different type of CCD. That we were able to, given our large cohort, we were able to put into summarize chart like this one. So I want to end with our prediction analysis. So we're trying to use machine learning to predict outcome associated with CCD based on the imaging and also genetic feature. And we were able to obtain 84% accuracy in predicting a more severe GDD when we consider brain anatomies together with a genetic diagnosis. So in summary, through comprehensive variant analysis, we were able to refine the genetic causes of CCD, expand on genotype-phenotype correlation, and advance our understanding of CCD. And again, we are continuously striving to uncover all genetic causes of CCD. Future directions, we use all genetic tools and we use all the newest genetic tools to try to identify even more CCD genes and pathways. In particular, we're paying a lot of attention to all those VUSs, the variants that don't have a classification yet. We're trying to understand the importance of intronic variants, so the ones that are not in the exons that we typically sequence, and the importance of mosaic mutation as well as in some inherited mutation. We use MRIs to help understand and predict outcomes for children with CCD, and occasionally we use in vitro and animal models to begin to approach targeted therapies. So we are continuously enrolling research participants, and in that is involved providing a saliva sample and sharing medical records and imaging and filling out a survey question at home. Anybody with CCD can participate, and both parents will be asked to participate if available. So to participate, you can contact our team or fill out a web-based intake form. Here is our contact information, but feel free to email me or Dr. Shear or Carolyn Lee. Finally, thank you so much to our past, current, and future research participants that allow us to do this work.